John Semple with Why Did I Get ITP? John.
Well, at least I wasn't called elderly. Hi, everybody. Yeah, I was told very distinctly by Professor Newland, no more military talk, so that's fine. So I thought I'd give you an overview of you have ITP, why did you get it? And the way to talk about that is to probably give an overview of autoimmunity in general. So I'm going to start by showing you this cartoon. And in fact, our immune system is composed of two basic components. On the left is what's called the innate immune system. And it's composed of anatomical barriers. It's got phagocytes in it, like neutrophils and macrophages. Platelets should rightly be placed in this system because they interact with the cells of this system quite regularly. Complements in this system, the only lymphocyte in this system is the natural killer cell, as it's called. And this system is always on. And it is really responsible for getting rid of most of the infections that we ever encounter in our lifetime. On the right is the adaptive immune system, and this is only composed of the two major lymphocytes, the B cells, which will go on to differentiate into what are called plasma cells to make antibodies, and the T cells, which orchestrate the antibody response, and they also have effector cells like what are called cytotoxic cells. For years, when I was a graduate student, people that did research in the innate system never talked to the adaptive people, and the adaptive people never talked to these people. And they figured it was separate systems and they had nothing to do with each other. Until, so it was considered that innate and adaptive immunity were separate entities. It was thought that adaptive immunity actually is the more important system of our immune system. But along came some pretty famous immunologists. And Charles Janeway, who was at Yale University, he actually formulated this hypothesis that really the adaptive system's pretty much off all the time. And it's actually really difficult to turn this system on. It's really hard to actually, for example, in a mouse, to immunize a mouse and make that mouse make antibodies. You have to trick the immune system into doing it by giving things called adjuvants. So he suggested that the innate immune system, in fact, instructs the adaptive immune system. And we now know that this is true. The innate system is actually the more important of the two, and it's responsible for getting rid of 99% of all the infections that we would require. So really, to adaptively respond, that is to make antibodies, you really have to be told, the adaptive system has to be told by the innate system, which has what's called genome-encoded receptors, and we'll talk a little bit about that. And then a little bit later, this very famous woman, Polly Matzinger, came out and suggested this danger hypothesis. And she said that basically our immune system doesn't really respond to foreignness, that is infections and that, but it responds to danger, such as bad cell death or something that's wrong, even a surgical scalpel knife cutting through tissues, things like that. Well, immunologists are pretty quirky people, and no better way of explaining that to you. To work in the immune system is not the easiest thing in the world. But this is Charles Janeway, very famous immunologist. Unfortunately, he passed in 2003, but he comes from a very long pedigree. His father was the physician-in-chief at the Boston Children's Hospital. His grandfather was the first full-time professor at Johns Hopkins, and his great-grandfather was the first New York Health Commissioner. Really amazing pedigree of physicians. This is Polly Matzinger on the right, and this is her dog, Mirkwood. And she's a really amazing person, too. In the 70s, she was a Playboy bunny in Denver. And then she was a bar waitress, and that's how she actually found her PhD supervisor, Robert Schwab. She's an accomplished jazz musician. She's a carpenter for Habitat for Humanity. She worked with Jimmy Carter on a lot of the building projects. She's a dog trainer. She represented the US at the 2005 World Sheepdog Finals, and this is Mirkwood, her sheepdog. And she now heads up the NIH Allergy and Infectious Disease Labs in Washington, and she calls it the GHOST Lab. Well, Polly, being the person that she was, She wrote a paper in the Journal of Experimental Medicine back in 1978, and she put Mirkwood, her dog, as an author on this paper. So this is the first published canine immunologist. And the reason why she did it in interviewed, she said he was the only one in the lab with me day in and day out. In those days, they brought dogs into the lab. I talked with him about the experiments, and he always looked at me with no judgment. Well, Henry Kunkel, the editor, banned her from ever publishing in the JX Med again until he died, and that was true. But after he died, she started publishing in JX Med again. So this is the type of quirkiness that immunologists really are. So I'm going to talk about autoimmunity, and I really hope that it's not a horror movie for any of you. So the concept of autoimmunity comes from this gentleman here, a very famous immunologist back in the early 1900s, Paul Ehrlich. And he literally talked about the horror of self-reactivity. That's the immune system that reacts against ourselves. And in fact, he's considered the father of immunology, and he coined this term horror autotoxicus, which really he was talking about with autoimmunity. He won the Nobel Prize in 1908 for his side chain theory of antibody development. He shared it with Eli Minichkov, who was the father of phagocytosis, which how our platelets are mainly destroyed. He cured syphilis in 1909, and he invented the terms chemotherapy and the magic bullet, and he standardized anti-serum. So an amazing individual. Now, autoimmunity is simply this. It's the immune reactivity against self. And by self, we don't mean psychological self. We mean a body part and antigens on our tissues. Now, there's a really important concept that you all need to know. We're all autoimmune by nature. Every single one of us, all mammals on Earth, have autoreactive lymphocytes that circulate in their body. But only in humans, only 5% of us are going to get an autoimmune disease. And the real question is, and we still don't know all the answers, is why don't the other 95% become autoimmune? And we're probably autoimmune because of the way our lymphocytes are formed in the bone marrow and in the thymus. But it might be a beneficial effect against cancer cells that develop. We have a little bit of autoimmunity that we can kill off some of these cancer cells that are also self cells. And the reason why we don't all get autoimmune is we have these complicated systems in our body, which are called tolerance systems. And there's central tolerance that occurs in the thymus, which is a gland just above our heart under the breastbone. And the thymus instructs all of these autoreactive T cells, it deletes them. And it's pretty good at deleting them, but it releases all the T cells that don't really respond to our cells, but are going to respond to an infectious agent from outside. But because it's not 100%, nothing is 100% in biology and nature except for death. Some of these autoreactive T cells do get out into the circulation, and I can take your blood and I can measure antithyroid antibodies in your body. I can measure all sorts of autoreactive substances in your body. But yet, most of us don't become autoimmune because we have other systems in the periphery that will shut off these autoreactive T cells and B cells. And I'm not going to go through all of these. It's just that these tolerance mechanisms are really important in keeping us from becoming autoimmune, but allowing us to respond to, for example, infectious agents. So there's many autoimmune diseases. It causes tissue damage. It leads to chronic disease. It's the third most common cause of chronic disease in Western society. About 5% of Western populations have an autoimmune disease. It's got a huge economic burden on society because people are away or at home, they can't work. And it's classed into either organ-specific or systemic. The organ-specific diseases are generally specific against a particular organ. ITP is an organ-specific autoimmune disease because only really the platelets are targeted. Insulin-dependent diabetes is organ-specific because it only targets the beta cells in our pancreas. Multiple sclerosis, the myelin sheath on our nerves. And then there's the systemic diseases that tend to be really diffuse. Lupus, for example, is a systemic autoimmune disease. It tends to affect many organs, and there's many different aspects of it that tend to be widespread throughout our bodies. So the requirements to become autoimmune, there's probably at least 3, but there may be others too. First, there's genes that are involved in autoimmune disease. There is a genetic component to autoimmunity. There's an environmental aspect of autoimmunity, which I think is really important. And then there's a whole aspect of internally, we have problems in our immune system. We have a dysregulation, as it's called. And when all of these come together, this is possibly why autoimmune disease occurs. So we're going to talk about these 3 things and just go over them to give you an idea of how this can happen. So we know that in autoimmune disease, multiple genes are involved. It's not due to one specific gene, like a mutation that occurs, for example, in hemophilia, mutation within factor VIII. Several genes increase the vulnerability or susceptibility in autoimmune disease. And how do we know that there's a genetic basis of autoimmune disease? Well, first off, in autoimmune diseases, there tends to be immune abnormalities that run in the families of people that have autoimmune disease. Not the same autoimmune disease, but if you have an autoimmune disease, maybe an uncle or a grandparent or that had something else, had an inflammatory disorder or some other autoimmune-like disease. So it tends to run in families, and that's the first clue that it might be genetic. The other thing is that there've been done studies on twins, identical twins, and tried to understand what the concordance rate is, as it's called. Do both twins get an autoimmune disease if they're going to get one, or does only one twin get it? And it turns out that studying these little fellas here, in fact, it probably represents that about most autoimmunity, about half the risks of autoimmunity are due to genetics. So it's a pretty strong component of autoimmune diseases. In and one group of genetic factors is really important. And I'm just going to talk to you. These are called the major histocompatibility complex. These are genes that code for proteins called HLA. You may have heard of that. These are the famous transplantation antigens. We can't transplant a kidney from myself into one of you because my HLA is different from yours, and your immune system will recognize that and reject the kidney. That's why we have to give kidney or transplant recipients heavy-duty immunosuppression like cyclosporin and that to shut their immune system down so they don't respond to these HLA antigens. And basically these HLA antigens, their main job is to present antigens to T cells. T cells have receptors that recognize these antigens, and they actually look kind of like a hot dog with a groove in it, and there's a peptide that sits in this groove, which is like the wiener itself. And it's the T cell receptor that looks down on this and can recognize it. And this is really probably a very important aspect of autoimmunity. These molecules play a role somehow in the fact that they present antigens to our T cells, maybe in an abnormal way, and they generate an autoimmune response. In ITP, there's been many studies that have looked at single mutations or polymorphisms at single nucleotide base, just one nucleotide base within the different aspects of genes. People have looked at immune molecules like cytokines and chemokines and found polymorphisms in many of these. In FC receptors, Dr. Cooter told you about those. HLA class 1, there's been lots of polymorphisms looked at that. And mRNAs, which are a new type of discovered a genetic component that certainly have shown polymorphisms. The problem is we still haven't figured out what these polymorphisms mean. We don't have a real good handle. They're there and they're associated with the disease in ITP, but we don't know if they're the real cause or not. And we have to do more studies in genetic studies in patients with ITP to try to understand this, because I think this is where we're going to get biomarkers, as they're called. We'll be able to potentially have a test for ITP, maybe based on maybe some genetics that we find. And we need to still work on this. Now, the other thing about autoimmune disease, there tends to be a sex basis in autoimmunity. Females tend to be much more affected than males for all autoimmune diseases. And the theories for this is that estrogen, the female hormone, tends to be immune stimulating, whereas testosterone may be immunosuppressive in some ways. So females get more autoimmunity. The dose of the X chromosomes may play a role, and females are certainly microchimeras from not only from pregnancy, but they're also from their parental cells. So it may very well be that these are the reasons why females get more autoimmunity than males do. In ITP, it actually pans out that that's kind of true. In very young children, males tend to be more affected in 1 to 5-year-old ITP patients, and then it equals out. And then during the childbearing years, females are predominantly affected with ITP. And then as we go to older again, the elderly, like Drew and myself, more males are affected with ITP. So there tends to be a sex basis even in ITP. Now let's move to the environment. We're almost going through and almost finished. There are many environmental factors that are associated with and may trigger autoimmunity. And probably the most important one are somehow infections. Somehow, and especially you people that may have children, young children with ITP, many times newly diagnosed ITP in children, the history turns up that there was an infection maybe 3 or 5 weeks before the child came in with petechiae and that. And that tends to occur in a big percentage of children with ITP. And so toxins can certainly lead to autoimmunity, or chemicals. Certain drugs can turn on autoimmune mechanisms. Certain foods can do it. And of course, you've heard about vaccines, very low incidence, but certain vaccines can induce autoimmunity and can induce ITP, for example. But these are very rare cases. So the environment plays a role. And to give you an idea, as I said, this is VZV. This is a varicella zoster virus. This is the virus that's responsible for chickenpox. Many children that have ITP, they have had a history of chickenpox just before they came into the hospital with their symptoms. And in fact, we know from work from our lab a long time ago had showed that the antibodies that the body's immune system generates against the viral infection, these antibodies cross-react. There's something on the platelet that kind of maybe looks like something on the virus. And so these antibodies cross-react. And then when the virus infection gets cleared from the child, these antibodies tend to go away. And in fact, the platelet counts come back up. And that might be why a lot of children will spontaneously remit for their ITP. But certainly there's infectious aspects to even ITP. Now a little bit about immune dysregulation. I'm almost finished. Something's wrong with the immune system. And all autoimmune diseases, including ITP, have multiple immune abnormalities that tends to correlate with the disease itself. The major question that we're trying to figure out is, are these abnormalities inherent? Were we born with them? Or are they due to some sort of result of the immune system being abnormally stimulated by, like I just said, environmental factors? And that's what we're trying to understand. So one of the problems that we know of in patients with ITP is this whole loss of tolerance. And that's true in almost all organ-specific autoimmune diseases. These cells here, you don't really have to know much about them, but these are really important cells, especially these things called Tregs, T regulatory cells. They tend to be reduced in patients that have ITP, and they don't work properly. And these are all the cell types that keep our autoreactive cells in check. And like in all autoimmune diseases, in patients with ITP, there's a deficiency of these tolerance mechanisms, and that's probably what allows us to become autoimmune. And we still don't know the reasons for these deficiencies, but there's a lot of organs involved in making them. The bone marrow is important because the bone marrow is a source of stem cells that may be abnormal producing these cells. And the thymus is important in these, particularly in these T regulatory cells that are faulty. So this lack of tolerance is probably responsible for why people get ITP. Now, this lack of tolerance, this lack of a suppression on autoreactive sets up a really complicated scenario. And this is what we know right now about how ITP works. And there's going to be a quiz at the end so that that you're going to know. But this is pretty well what we know about ITP. And I tried to put this up to put everything that we know about ITP in terms of how the abnormalities really work. There's hundreds of papers that have been published on this, particularly in these cell types here and in these cell types in the middle. But nonetheless, antibodies are generated from this big mess. Then these antibodies bind to the platelets and they're either destroyed by macrophages or they might be able to fix complement in the plasma and kill the platelets. And there's CTLs or CD8 positive T cells that are generated. Nikki Cooper has been working on these cells, particularly in the spleen. And these cells can move into the bone marrow and they can destroy megakaryocytes. And that's why, presumably, there's a production problem in ITP. So this is what is my world, and this is what we try to work on and try to understand this. But this has really become important because Big Pharma has now been looking at these scenarios, and they're trying to develop drugs that hit each aspect of these. But, you know, there's all sorts of drugs that you're going to hear about that affect various parts of this scenario. TPORAs work on these cells here, the megakaryocytes, and even though they're being destroyed, TPORA makes them resistant to the death, the destruction, and also turns them on, and you make more of them with TPORA, for example. Steroids work basically on many of these cells and generally suppresses the whole milieu. The problem with steroids, of course, are the side effects. And if you take them too long, you start suppressing the immune system so that you might be prone to infections. So in conclusion, we are all autoimmune by nature. It's important to remember that, but only 5% of us will become autoimmune. ITP is primarily due to this lack of self-tolerance in these suppressive systems that will turn off our autoreactive clones. Genetic factor and environmental factors are really important in playing a part in whether we'll become autoimmune. And understanding these types of things will help people like me in that to identify perhaps targets for therapy in the control of this autoimmunity. So I just want to thank the people in my lab and my collaborators around the world for helping us do the things that we do. And I thank you, and I'd be glad to take any questions. Thanks.
Thank you, John. As you've noticed, my son's allowed to grow old in John's lab. If there are any quick questions, we could take those. Otherwise, we can incorporate any questions. There are one or two, I know, within the Q&A at the end. Stunned into silence.
Yeah. Either completely clear or it's complete mud.
Clear as mud. Thank you, John. That's excellent. Oh, Fred, did you want to ask? Well, Vicky's just plugging in.
You think mud?
Amorous, we use as a biomarker.
Amorous.
Amorous.
Tamarind. Oh, tamarind. I don't, you know, potentially, but I mean, we've only just discovered these in ITP. I think they have to be worked. We don't